All right, welcome back to InnoFX Live. We're here today with Betty Howard, radiation team member and manager at STERIS. So Betty, terminal sterilization, back to basics. I use that simply because I think people need to understand all of the aspects. So let's just, what does it mean? I'm going to label my product sterile. So I just sterilize it and I'm good to go, or how's that work?
Well, see, that's probably the biggest misconception. People think the act of applying a sterilizing method automatically means the product is sterile and is sterile forever. What you have to do is you have to match the modality that you're going to use to give the level of assurance that you want that that product is sterile, and balance that with that the product is still going to be as functional as you need it to be and not have any degradation products or damage to it that would impact its stability or its function. So it's a balancing act. You want to make sure you make it as clean as possible and that you meet all the regulations that are requiring it to be labeled. You can't just label something sterile because you did one quick test to it. You have, for every modality you use a sterilization process for, you have requirements for how you have to monitor it and count it so that you know that that product can be labeled sterile and meet all regulations for sterile labeled products, regardless of what kind of product it is.
I think in a lot of cases, people feel that it's an end game, sterilization, and it makes up for all the mistakes, or not mistakes, oversights. So walk me through it. I want to make a pick a device. It has to be sterile. What's my best approach?
The absolute best advice I can give anybody is to consider sterilization at the very beginning of product development. When you're developing a product, you bring together engineers, you bring together people who are chemists, you bring together frequently even the marketing people and the doctors and such that will use it, because you want to design something for use. Well, at that time, you should also be designing for the sterilization process, because that's going to come much later. The decisions you make early on can damage your ability to effectively sterilize, or can make your sterilization process much harder to do, much more costly, and much more narrow focused, where it makes it more difficult to have efficiency. If you're thinking up front about, okay, which materials do I use? What am I thinking about sterilizing it? Am I thinking of using something that involves heat? Am I thinking of using something that involves high energy, like a radiation process? You can select good materials to make those successful. Your first guess may not be the perfect one, but at least if you're thinking of it up front, you avoid some of the biggest mistakes people will make. So if you're designing for function, part of that function is to make it sterile. So if you start from the very beginning, I'm a big fan of just having somebody in the group in the development growth going, hey, what happens when we sterilize that? Just to keep that focus on that at the end, it has to be sterilized. The saddest thing I see is when somebody does what you described. They've worked on something for a decade, and they put it in a beautiful package, and everybody's all excited about it. It's going to serve a wonderful function, and they run across something that is a little different than they've had before, and all of a sudden they go, oh, we'll just sterilize it. Well, in fact, what you're doing is you're not sterilizing, you're sterilizing in quotes. You're applying a sterilizing agent without the control for the sterilization process. To call something sterile, you have to have process control right from the beginning. You want to have a product that you make reproducibly so that you get a reproducible result when you make it, as well as when you sterilize it. So it starts from the very beginning. It's the basic science that you use in developing the product and finding a need for it also applies to the sterilization process.
Well, let's look at it. Let's look at it a couple of different things. I think one of the things I always found is guys who come in and they decide that they're going to make a sterile product from an NCE. So, what in the synthetic process should these guys be looking for? Obviously, if the end game is to make a sterile product, so I'm a synthetic chemist. What do you think I should be thinking about?
I think you should be thinking about the matrix you're going to put it in, because that can have a huge impact. If the matrix it's in is not less, necessarily less stable, but creates byproducts that are going to cause problems with the actual active material once you've sterilized it. You have to consider that. You have to consider the format it's going to be in. Will it be in liquid form? Will it be in some sort of emulsion? Will it be in lyophilized form? All of them have advantages, but one or the other may provide a more stable, effective material. So sometimes there's the desired route, and then there's the one that's the best benefit to the product. So you start from the very beginning looking at those things. The other thing is you look structurally at molecules. There are certain things that are known that will be interactive with certain sterilants. For example, there's some structures that are known to be very stable when you do irradiation. There are others that are known to be very unstable when you do a process that involves heat or moisture. If you look for those things, you can eliminate certain kinds of sterilizations because you know that they're automatically going to impact the active material.
So, having said that, so when do you think giving thought to a sterilization validation comes into play?
Okay. When you have you should not validate a sterilization process until you have process control for making the product that you want to sterilize. Because if you have a variable coming in, you're going to have a variable going out. Most sterilization processes are very robust. There's a lot of degree of overkill in them. They can handle some variation, but they can't handle continuous randomness and still give you the end result. And by end result, I'm saying not just if I did a quick sterility test, would it pass or fail? I'm saying a level of probability that it will be sterile, which in most sterilization languages, you're referring to things like a 10 to the minus 6 sterility assurance level or something like that, where there is a known amount that people are looking for to have that sterile claim and be able to put it on the label. And it's all a controlled environment that you're making the product in, and you keep doing the same thing over and over. You should not introduce new organisms that weren't there before, which means you haven't introduced new resistance to the sterilization process. And you start from the very beginning. You validate a sterilization process when you have a process that's in control. That's the only way to be absolutely successful.
All right, so let's just think for a minute. You know, there's a lot of things that people don't always think about having to be sterilized.
Right.
A lot of times inhalation products, you know, it was, you know, all of a sudden, wow, geez, I didn't think I had to have that sterilized. Mm-hmm. Medical gases, for example, there's another one. So, I mean, what is the common approach here? Let's stick with medical gases, just as an example.
That's probably the one I have the least experience with. I have seen some of the CO2 cartridges and those types of things, but the inhalants, I haven't really worked with much. But the same thing would apply to them. They are in a state where they are vulnerable to a chemical process or a process that could alter them. You have to understand the modality that you're working with, but also the path of synthesis for the product that you're working with, because then you know where the points of concern are, or maybe a point at where you would have to protect a molecule or a device. If you know the mode of action of something, you can usually predict where the weak points will be, and you can intervene to try to protect them.
I see. Mm-hmm. So it really comes down to where do I sterilize it?
I think it's how you sterilize.
So
there's a lot of ways to sterilize. That's the beauty of sterilization. It's been around since, I mean, think back to Campbell's soup and things like that, where you had, you know, the concern for botulism or something in food products. From everything we've learned over the decades, centuries at this point, we've learned a great deal of ways to kill effectively microorganisms. And what we've done as industries, we've learned better ways to deliver those methods so that they can be done quickly, efficiently, relatively low cost, and very reproducible. So you have many more choices now than you ever had in the past. It used to be there were only maybe a handful of things. You're still seeing new entities coming out as ways to modify bacterial or viral populations. So you have a lot of choices. That's why you have to look at how a terminal sterilization process will match the needs and the physical nature of the product you're trying to sterilize. And make good decisions up front so that you have lots of choices. The more choices you have, the farther you'll go. Because I've rarely seen any product, go through development and not have a glitch somewhere along the way, where you had to make a veer somewhere and to modify. If you're thinking of options as you're going in development, then those changes that may happen don't knock you off your path. They just give you another path to follow.
So, sounds to me like I have to have a formulation, a package, and a process pretty much worked out, and then see how the sterilization works with that. Is that
I would
pretty
say
much it?
absolutely the formulation. You have many choices that you can modify a package much more readily than you can the formulation. So if you have a final formulation, the way you will sterilize it, that's what you want to test. Now, it's not a bad idea to, in development, as you're developing that formulation, to look at variations on it in an R&D level. See what impact, say, irradiating something has, or heating it has, or using some form of gas sterilization, so that you know what to look for, because that will tell you where these critical areas are in your structure and give you a chance before you're to the final one. Or maybe you come up with several variations and now you pick out which one's best based on not just the functionality of it, but also my ability to sterilize it and to keep it stable.
Let's talk about, things that you challenge, things that challenge a process. So one of the things that I think people seem to forget is inherent bioburden of things that they process. So I think part of that, so how does one come up with a strategy that balances, obviously you can't, you're not going to go out and buy it sterile, right? However, you also have to have it such that, and you were kind enough to explain it to me, that when we do sterilize it, there's got to be not that many dead bodies in it in the end, especially if we're going to make a claim. So, so what has been your experience with that? Obviously you don't want to spec yourself out of existence, but on the same token, you don't want to challenge your process. So how do you work that out?
True. The, if you look at pharmaceuticals, by their very nature when they're made, they're usually very pure compounds. Making them pure also impacts the total bioburden or the contamination level in it. By doing that, you make your job in sterilizing much easier because you've, maybe in some cases dumb luck, before you even thought about sterilization, you made the population that you're trying to target much smaller and much more uniform. You generally get raw materials from very select suppliers. You don't willy-nilly change suppliers, so you're getting it from the same source material, same vendor, same location it's coming from, so one would expect the bioburden to be reproducible. By the purification process, you tend to lower the population you're working with, which gives you a much smaller targeted population to try to eliminate. You can also now, and even at the R&D level, start looking at what the composition of that population is. What types of organisms are present? Are there any I have to be exceptionally concerned about? If they're common things from handling, a staph or something like that are very easy to kill, you don't worry. But what if you get something in there that is known to be pathogenic? You want to know if it's there. You want to know how you have to be able to eliminate it. So you're developing in your R&D process knowledge of not just the function, but of the materials you're working with. And that will always be valuable to you when you're trying to reduce your bioburden to a acceptable level or completely eliminate it by labeling it sterile.
Okay, so I think that's part of what we're going to do. Now we heard about the raw materials. On the back end, okay, what do you need to consider as far as the method in which you're going to terminally sterilize? I think we've heard about the modality has to be such that it doesn't compromise the product, but what else? What else do we have to consider?
Okay, you're going to take into consideration the format the product is in, because some will immediately eliminate certain forms of sterilization. For example, if your product has to be in some kind of container that has absolutely no permeability to gas, that'll make it difficult to use a gas sterilant because you're never going to get it in there. If your product is in liquid matrices, there are other things that will not be able to be done because you might dissolve the sterilant in it and not be able to get it back out. You'll learn all these things as you're going along in development. Say you have a product that you know to be sensitive to heat. You have to look at the modalities that are out there for sterilization and say, do any of these raise the temperature of my product above a level that I think my product will remain stable? If it does, and it's still the best method out there, what can I do to make sure that that temperature during the sterilization process doesn't exceed the part where I have a problem? It's not impossible to package things in, say, under cool conditions where they're surrounded by ice or cool packs, or you learn that they can be exposed to a certain temperature for up to a certain amount of time. So you can judge the modality, the physical natures of the modality in sterilization. Those don't really change much. You have whatever they are, but you can modify how you deliver them. You can modify how you present your product to them and try to find the one that gives you the best balance. But in many cases, it will be trial and error until you know. But all the information you know about the product that you got out of development will help you make good decisions, so that you'll eliminate a lot of the possibilities of what's out there that could effectively sterilize it but create a pile of useless material.
We had an interesting discussion. One of our first presentations was on modular manufacturing. The facility is no longer a facility. Pretty much what it means is the stuff is contained in a pod. We could pick the pod up and ship it anywhere. It's particularly popular with biopharmaceuticals. What I found lacking, at least in the discussion we were having, was the aseptic handling, filling and sealing it. What has been your experience there? I mean, it seems to me you have, the resident flora. I mean, you're going to take it from point A and move it to point B. C, I mean, how do they go about identifying this if they're going to do something like that?
The most common, the best thing you can do in those cases is when you move an entire manufacturing process, realize that you have not just drop shipped the whole environment with it. You have to now requalify that whole thing as if it was new. You've put it in a new place, you have new people, you have new cleaning procedures potentially, but you have to make sure it still produces what you thought it did and does so the same way. The nice thing about most of those aseptic type processes is they have very robust protection mechanisms to maintain sterility in that room, because the aseptic process doesn't make it sterile. It's what you bring in. So that's actually one of the benefits to a terminal sterilization process, even with an aseptic fill, is that you can sterilize, document the sterility of all the stuff you bring into the aseptic process, so you protect that process from contamination, or where you would, I mean, a minor contamination in an aseptic process would mean the whole thing shuts down and have to be completely taken apart and cleaned again before it could be used. So that bringing in, using all of the tools in your toolbox, basically, instead of, sometimes it's all not start and finish, we'll do everything completely terminal. It may be a terminal sterilization process is what gets you into the aseptic process as well. And I actually am seeing a lot more of that, because it can speed up the process of developing a process for making something totally aseptically.
I think one of the most interesting things is the inevitable particles within a vial. All right, and and customarily, I think people fail to realize that the cyclic nature of the temperature actually creates the situation. I think you and I had talked about this the other day. Invariably, they don't start with the base ground stable form. What has been your experience in just speaking about it? It may be a war story, but it seems to me that a lot of people invariably, and they just keep adding on stuff. They got crystals, well then they add on a filter, and then they add on it. I mean, who are they kidding?
You lose more material that way, and you end up with less and less of what you really wanted. If you're looking at the synthesis process up front, you probably correct for a heck of a lot of that stuff so that you don't. There's the big fear is at the end that you won't have the particle sizes you want or the activity you want, and you tend to look at each one of those things one at a time when the crisis comes up, as opposed to controlling the manufacturing process from the beginning, so that you're producing something that's within your specifications. The other one is realize that specifications can never be pinpoint. They always have to have some range with them, and you have to know that the range over which you find it acceptable will tolerate what you're going to use it for. And you're right, very, very often I will get calls where somebody will want to correct something that maybe just happened in one batch. And that's very difficult to do because you don't know what caused it. You also don't know what the intervention at the end will do to everything else that was done before.
In your experience, just one of the things I've always found interesting is that a process may not be fully integrated. In other words, they make it here, fill it here, but sterilize it someplace else. Mm-hmm. That's got to be a tough one.
As long as you do the same thing each time, if you always make it here, package it there, sterilize it there, you're accounting for all that variability. That's part of having that production process in control. It doesn't have to be always a perfect system. I mean, it would be nice if it could be. But if it's in a controlled system, you know what your enemy is. You know what you have to fight. You control each of those processes as best you can. So your comment about modular, you can look at the people who are in charge of this part. They can make that the best they can, and as long as they don't change, and then you go to the next one and they don't change. You have to constantly monitor this to make sure things are not changing. But in terms of for a sterilization process, that's acceptable because you've accounted for all of the possible changes in bioburden. You've accounted for the different changes in the production process because they are natural to that process. I mean, would you like to get rid of them? Always. The less variables you have, the less possibility of change. But if it's reproducible, what you produce, going through all of the steps, timing and such, the temperature range over which it will be processed, at what point you intervene, at what point you call it an action level before, or something where you have to do something really severe to look at it. You have to know your process well enough to know when change is significant and monitor it. And then in that case, then terminal sterilization can be a general cleanup because it was set, the conditions for that terminal sterilization were set, assuming all of this happened beforehand. So it still works, but it's all about process control. You control the process, means you control the bioburden in type of organisms present, the quantity of them, and by controlling the type of organisms, you control their resistance to the sterilization process. So it works. I've seen some process where I couldn't believe that people had as many steps in them as they do. They do it because of efficiency, they do it because of cost, they do it because of volume that they can produce, and it works. It works because each step has to be controlled. As long as you control the process in, you control the process going out.
Is there a single go-to terminal sterilization approach for anybody? I mean, I'm sure you've been asked this question 100 times.
Well, I have to admit I'm a little jaded because my largest area of expertise is radiation. So I'm willing to work really hard to make a radiation process work, but I work in a company that has multiple modalities. The reason why multiple sterilization modalities exist is that there isn't one that will work for everything. You pick ones, you many times have to match the product's needs to the sterilization process. I mean, many times you can get a great many of your products in one, but there will always someday be one product that just doesn't fit the mold. But it's such a valuable product to you that you need it to be sterilized. So you want to be open to other possibilities. I learn every day more and more about the modalities that I don't see every day. Just knowing what they can do in very unique situations. That's why you want to always keep your mind open to the possibilities. Understand how they each work so that you can always relate them to your process. Use all of them if need be. If you can it's a great tool to be able to get all of your products doing the exact same thing, but that means that all your products have some parts of them that are pretty similar. If they're very, very unique, and which probably makes them valuable to you as a company, you may have to make adjustments to do what's best for that product. So I try not to be prejudiced.
Well, that's okay. We accept prejudice,
okay.
But
well, like I said, I do tend to.
Well, but again, I've been in a situation where we were working on a very unique combination product.
Mm-hmm. Yeah.
And they went to their go-to sterilization process. You and I talked about this a few days ago. Mm-hmm. And what blows my mind is it happens. It's a polymer they used before, and they get a different molecular fraction
Okay,
after
yeah,
they
that's
bombard
common.
this thing. And it's like, and now they're saying, well, it's still okay.
Says
Well,
who?
how do you know that? But you
Yeah.
didn't put the spec in the beginning.
Yeah.
I mean,
Yeah.
how many times do people reinvent the wheel? I mean, that.
Yeah. Well, I mean, the example we talked about is many times you buy something, a component that's going to go into a product, and you buy it based on its molecular weight without looking at the label of how it got that molecular weight. There's a lot of ways to get a molecular weight average of a certain number. If you end up with something that has, 50% of something very, very low and something 50% of it very high, you get a molecular average somewhere in the middle. But if you're looking for your functionality and the stability and the use of the product for it to be pretty close to that median number, you're not going to get it if you've got these things that are dramatically different. So you're back to controlling the quality of what you put in.
And I think in a lot of cases, I think the problem they get into is they try to explain it away as not being a functional defect. I never quite got
Sometimes
what
it's not.
non-functional defect meant.
Right.
Defect's a defect. You know?
Okay. I look at that as, does it meet its specified purpose? Is there anything about it that is now unsafe or less safe? Is there anything about it that makes it less stable in use? That's one of the first things you decide, I think, in a product, what your requirements for that product are. Not necessarily what the end user's requirements are. What are you promising this product will do? And you have to do everything in your development to make sure that you meet the specifications you say you will. I mean, I think sometimes people start out with very, very narrow specifications because they don't know enough about their product, and that's where your R&D work comes in. You learn more and more, and you learn that, okay, I like it right here, but it works just as well if it's a little under, a little over. And now you just set your limits. You now know that as long as it's within there, I can meet my specs. If it falls out, you give it up. You say, okay, that's not meeting my specs. I either have to change my specs, which could be bad, or I accept that that lot or whatever does not meet my specs, and that you can't go back at the end and do something to try to fix it. It has to be engineered up front.
Is it reasonable? Is it reasonable to assume that if I have a polymer in this system that I'm using, and again, polymer covers a lot of
Yeah, I know.
ground, but is it reasonable to expect molecular component change, in generally speaking? Because
You expect structural change somewhere. For example, a polymer could cross-link when it's sterilized. It could break down into smaller components that are the same material but smaller sections. If you do that, maybe your viscosity changes when you do that. It's still chemically the same thing, but its behavior is not what you intended it to be. So you do have to look at that. You have to look at whether or not, the general rule is almost everything does one of three things. It either breaks it down, cuts it, scission, it cross-links it, double bonds where there weren't double bonds, or something happens to it and it goes right back where it started, recombination. Unfortunately, the world does not usually go totally to recombination, because then everything would be good in sterilization and manufacture. You have to know which way it tends to lean and know what those byproducts might be. Because if you do, you can deal with them. Or choose another modality because it didn't give you what you needed.
how about processes which allegedly are self-sterilizing? I mean, I've heard this before.
I have too.
You know?
I still see, I will tell you that I have had to terminally sterilize sterilants because you put them into, for example, a sterilant in a bottle. It has to have a certain amount of contact time to either disinfect or sterilize. You're now putting it in a bottle that wasn't sterile, and it may have organisms in it that are not as affected by the sterilant that you just thought was in that bottle. So it still comes down to knowing what the actions are. I don't see as many of the chemical disinfectants other than for surface treatments, but that's because I usually see finished goods in packages and boxes that somebody's going to ship directly from one of my facilities right to an end user. But I can see that if you think about, you put a rather severe chemical in something and you leave it sit for long enough for the action to occur, it should be self-sterilizing. But you have to figure out how to quantitate that as well. Or what is it acceptable? I'm positive there's probably regulations on that, the details of which I'm not particularly familiar with. But every mode of anything that's called sterilant has a way to document whether it's effective or not. And on a chemical, we'll have limitations on, how it's diluted, how long it has to make contact, how long it's good after it's been diluted, how long it's good after it's been opened in some cases if they're subject to oxidation. So you have to take all of those into effect too.
One of the other things, and this gets back to some of the other, especially in oncology research, most of the compounds we work with there have the solubility of sand. So what we find in a lot of cases, we wind up with a suspension.
Yeah.
So that's got to be particularly difficult to manufacture from our standpoint. I mean, suspensions are inherently unstable. That's the rules.
Yeah.
So what has been the customary approach in this? I mean, how you got double layer effect? I mean, so what's the is there a go-to sterilization?
I do see in publications that many things have been used. I personally see more that are done with radiation because the suspensions can't be filtered. So you want something you can do in finished form. Many times the suspension you put them in can be engineered to make them more stable. And the material that's in there, like you said, it's consistency of sand. It's not necessarily something that's very unstable structurally.
Right.
What you're doing is just making sure it stays in a format that can be delivered. In that case, the active part is usually the very stable part. It's making sure that the matrix is also stable. I do see quite a few of them, and actually even going back a few years where I don't even think we were looking at it. People were coming to us and saying, they'd already tested it. They're doing exactly what I'm telling everybody to do. Try things early on and see what works, because you do find that every active material or active structure, if it has to bind something or other, has very unique properties that you engineered into it. And it's what makes your product unique from somebody else's in some cases. So that just looking at something that might be in literature may not answer your question, or may tell you can't do something when you can. There is no substitute for trying these things. You have to know what really happens to your product, not somebody related's product or what you think their product is. Take all the information that's out there to make good decisions on materials and avoid things that are just obviously going to be problems. But once you get down to it and you start looking at things that could be your path forward, whether it's a device or a pharmaceutical or a suspension or a combination product, there's no substitute for actually looking at what you might produce. And do it early, before you have to make submissions to regulatory agencies. Because the worst thing that happens is people spend a decade or more developing a product for a wonderful purpose that serves, a need that's out there that's not met. And then reach the end and realize they have all this data that they're using for their submission to show safety, efficacy, and stuff, and then want to sterilize it at the end. The problem is that, for example, just in radiation, there's actually a line in the CFRs that say, there's a whole list of things that are considered a change to a product. And right in that list, and these CFRs have been around a long time, let's say irradiating a pharmaceutical entity means it could be a different entity. So they invite you to, well, they don't invite you, they require you to go there and prove that nothing bad happened to it. Because they know that these things can cause a change. Sometimes I get calls from things where the intervention of a sterilant made it better. So you never know. It could be a bad thing, but it could be a good thing. But you always are, the goal in pharmaceuticals is always not to get any degradation products. You don't know what they are.
Right.
And to keep the other ones that you know of down below a certain level.
Right.
As long as you can do that, your sterilization process is good too.
Right. And from what you're also telling me, a little ETO in my product never hurt, right?
True. That's the other one. Many times you see certain subsections of healthcare who won't use one process or another because they have a fear of it. I've actually gotten calls from people asking me, how much contact somebody can have with a product after I've irradiated it. Very brilliant people, but it's an area they don't understand. That is not radioactive, that the radioactive never touches it, but they don't understand that, so they're afraid of it, so they avoid that. With, for example, ethylene oxide, you're worried about the evil word residuals. Well, residuals can be controlled by having a good process to remove them. The gas goes in, you got to get it back out. If you can get it out effectively and your product is not such that it retains the bad parts of the gas or creates other residuals, and products that you don't want, you can control that. That's why those processes have all those variables to control.
Yeah. I remember we had a couple of natural products, components, and
those are always fun.
Yeah, but we irradiated them, much to our chagrin.
Yes.
But nevertheless.
See, I taught you said irradiate, you didn't say sterilize.
Right, no, we irradiated these. And what was interesting about it was that, other than changing the properties entirely, we were sitting around trying to figure out what exactly it was. And the guy asked the same question with residual. The guy I work with raises his hand. He says, well, if I shine a flashlight in the drum and turn it off, are you afraid that the light's still trapped in there?
I like that.
Yeah, I was surprised. I mean, but, I think a lot of people don't get it. I think what we were trying to do today in a lot of cases is that one of the things Betty and I talked about when we put the program together was people have a tendency to bolt this stuff on the back end.
Yeah.
Don't give a lot of thought to it. Worse yet, that it has to be part of the whole process development aspect. And we had talked about it from a quality by design standpoint. I think you heard Kay.
Yeah, I was going to say, I got the lead in from the previous speaker. It's perfect.
That was it. So I think we've about run out of time. So Betty?
I would say the take-home message is seek knowledge early. Don't be afraid to ask questions. I know that my organization, we have people who are there just to ask questions. So find out what you need to know. You may put it in the back of your head. You may not use it now, but it'll come back. It's another tool in the toolbox. So use them all.